The overall goal of this experiment is to assess the immunomodulatory properties of human mesenchymal stem cells, or MSCs.
This method can help elucidate the mechanisms involved in human mesenchymal stem cell immunomodulation, a process that may have therapeutic relevance. The main advantage of this technique is that both the occurrence of effector leukocyte proliferation and the number of cell divisions can be assessed. Demonstrating the procedure will be Pei-Ju, a technician from my lab.
To isolate PBMCs, begin by diluting 25 milliliters of a heparinized whole blood sample with 25 milliliters of PBS in a 50 milliliter conical tube. Next, add 15 milliliters of phycol pack density gradient to a new 50 milliliter tube. Then, holding the tube at an angle, slowly layer the diluted cell suspension over the density gradient, taking care that there is no mixing of the layers.
One of the most critical steps in this experiment is the layering of the blood without disturbing the density gradient. This is to ensure a high purity and low red blood cell contamination of the isolated PBMCs.
Now separate the cells by centrifugation. 3 distinct layers should be apparent: the upper plasma layer, the bottom phycol pack density gradient layer, and a thin middle PBMC layer. Using a Pasteur pipette, remove the upper layer, taking care not to aspirate the interphase layer. Then use a 10 milliliter pipette to carefully transfer the PBMCs to a 50 milliliter conical tube.